I'm a student, a PhD student in Dr. Ati Singh Lab, and it's my turn now to introduce Dr. Berger. So Dr. Berger is joining us this evening or afternoon from in Ames, but she's from Adelaide, Australia. Like she said, it's 8 a.m. Saturday. So thank you so much for joining us this early on your end. We appreciate it so much. So with a background in biotechnology and a PhD in molecular plant biology, Dr. Berger used a 2-year fellowship at the Australian Centre for Plant Functional Genomics, focus on the then emerging field of plant phenomics. Since 2010, Dr. Berger has worked at the Adelaide node of the Australian Plant Phenomics Facility, and in 2015 became scientific director. She enjoys the diversity of working in a national research facility with a whole range of different users, research questions, and challenges on a daily basis. Dr. Berger has focused on establishing protocols and methods for high throughput screening of various plant species in controlled environments, using imaging technology to help the research community take advantage of the benefits that modern phenotyping techniques have to offer. Our presentation today is titled Trait Dissection and Phenotyping to Study Complex Traits in Crops. Please join me virtually in welcoming Dr. Berger. The floor is yours, Dr. Berger. Thank you.
Thank you very much for the opportunity to present, and I appreciate that it's probably been a long day full of interesting presentations for you. Day's just starting for me. I should also apologize, I've got a pretty bad cold, so my voice might be giving out at some point. That's the disadvantage of living in a COVID-free state. You mingle and socialize and catch colds, but really can't complain about the place where I'm living. So yeah, as mentioned in the introduction, I work at the Australian Plant Phenomics Facility, the Adelaide node, and I hope I can manage to move my slides. Here we go. Just as a brief introduction, the Australian Plant Phenomics Facility is funded by the Federal Government of Australia under what is called the National Collaborative Research Infrastructure Strategy, or NCRIS. It means we are able to offer phenotyping services to researchers from across the country, but also internationally. So we are not limited to working with researchers here at the University of Adelaide. We are a truly national facility, and we are used both by the public but also by private research sector. In addition to the node here in Adelaide, we also have 2 nodes in Canberra, one at CSIRO and one at the Australian National University. And across the 3 nodes, we offer a whole range of different phenotyping services. That's everything from the controlled environment phenotyping to field phenotyping using UAVs and also manned aircrafts, sensors out in the field. And then we also have a team of experts helping with the analysis of the quite complex data that is generated by phenotyping tools, so that we can support the researchers through this process with their analysis. So what I want to talk about is how do we use phenotyping in research and breeding, and Francisco has already given an excellent overview of how that is done mainly in the field. I want to present something sort of slightly earlier in the research and pre-breeding pipeline, which is focused on the controlled environment. And quite often people ask, well, you know, what's the point of phenotyping in the controlled environment? It's not the field, and which is true. So what I want to do with this slide is just explain that phenotyping in controlled environment and the field, they serve 2 different purposes within the research and breeding pipeline. In the controlled environment, you can hone in on very specific traits and study them in isolation without all the G/E interactions that you have in the field. You can also do multiple runs per year. You can look at early germplasm where you only have a few seed available. and you can apply treatment at the individual plant level and apply a very precise treatment. But yes, obviously it's not the field. You study plants in isolation rather than in a canopy in a closed community. So your plant-to-plant interaction is sort of missing there. On the other side, the field, obviously, you are growing in the target environment of where you want to ultimately grow your fields later on. They are grown in a closed canopy. And as Francisco just showed, there is a whole multitude of sensors now available to phenotype directly in the field. There are also disadvantages though, and one is that, you know, one field and 1 year is not the same as the next. So you have large G/E effects. So you really need multi-year and multi-environment trials. And if you work very early in the breeding program where you just don't have enough seed available, that can become very difficult. So the two aren't contradictory or exclusive of each other. They are just often at different stages in the process. And this is a slightly different slide to what Francisco also presented with the physiological breeding approach. So what we are doing is really trying to use trait dissection to break down complex traits. So we are certainly not trying to mimic yield or select for yield in a greenhouse. That would be quite foolish to try. What we are trying to do is identify traits that we believe contribute to yield in the field. So some of those puzzle pieces that contribute to either higher yield potential or abiotic or biotic stress tolerance. Then identify genetic variation for those component traits, develop a robust screening protocol, and then ideally find the underlying genetics for those traits, and then study the relevance of those genetic markers and whether they are associated indeed with yield in the field. So I want to focus on salinity in this talk, mainly because I've worked on it for quite a while. But similar approaches would be relevant for other stresses like heat or drought, for example. But yeah, really salinity is a global issue, especially if you look at Australia. There's hardly any area in Australia that isn't affected by salinity, and these parts in Western Australia, South Australia, and the East Coast is primarily where cropping is done. So all of those are affected by either saline or sodic soils. And salinity has sort of two different types of effects. One is an immediate effect, which we sometimes call the shoot ion independent stress, or SID. Having said that, that name seems to change every once in a while because we still haven't found a name that sounds good and accurately describes what's happening. But pretty much as soon as plants are exposed to salinity, and in cereals, that's certainly primarily the sodium rather than the chloride, that leads to often stomatal closure, reduced leaf expansion, and overall reduced shoot growth. And that can often be measured within minutes, certainly within days. Longer term, once we have toxic levels of ions accumulate in the shoot, we then also have the shoot ion dependent stress, where we can have early senescence of and premature senescence of the older leaves. So the shoot ion independent stress often affects young leaves, whereas the shoot ion dependent stress generally affects the older leaves. So if we look at the individual traits of salinity tolerance, there is the shoot ion independent stress, tissue tolerance, the ability to tolerate high levels of sodium. It can also be sodium exclusion and leaf elongation, a whole range of different traits. We know quite a number of the genes for sodium exclusion, and the reason for that is it's very easy to phenotype. All you have to do is cut off a leaf, digest it in nitric acid, and use flame photometry, and you can do hundreds and hundreds of samples for, apart from the labor cost of someone doing it, not much money. So we've known quite a few of the genes involved in sodium uptake into the root, relocation into the shoot, and then also unloading of the xylem and potentially recirculation into the phloem. And yeah, that was really because it's easy to measure. However, if we look at the shoot iron independent tolerance, so the ability to maintain growth, we know a lot less. And the reason for that is it's a growth-related trait. You need measurement through time. And we just didn't have a good phenotyping protocol for this. So we really, needed to develop an assay to do that, to then move on with forward genetic screens, and then identify QTL that can be tested in the field. So what I want to present in this talk is a couple of different examples. One is from developing a protocol to measure that growth response, using it in forward genetics, and then also further dissection of that growth response, and then some examples where we moved all the way from trait discovery to field trials. So this slide is actually data that I generated back in 2008 when I started my postdoc. At that time, we didn't have the plant accelerator. I was using a manual imaging system, and I was growing barley plants in a hydroponic system because it was easy to maintain the sodium concentrations in the solution. And really what I did was grow plants and image them over time. You can see a couple of breaks. That was probably the weekends where I then got sick of coming in every single day. But certainly that phase after salt application is where I try to have daily imaging. And then very simplistically, what I did was fit an exponential growth curve through those early growth responses, looking at the growth rate in control and the growth rate in salt to get an idea of how much growth is reduced. And then the hope was to find genetic variation in that trait. And if we look at these two barley lines here, Clipper, which is an old Australian line, Sahara, more of a landrace, then really the difference wasn't all that great, for example, similar for the others. So we're looking at the ratio of relative growth rate in control to relative growth rate in salt. Sorry, the other way around. And there wasn't much there, which was slightly disheartening. However, by the time this was happening, the plant accelerator was being built. And what we did, we moved from a hydroponics-based system to a soil or soil substrate-based system. And if you look at those same two lines, all of a sudden you can see quite different responses where Clipper is really falling over pretty much when high levels of salinity are added to the soil compared to Sahara, which seems to sort of still power along fairly well. So blue is the control, brown the 150 millimolar sodium chloride, and red 250 millimolar sodium chloride. And Clipper pretty much dies by the end. The other thing that this obviously shows is using an exponential growth curve in those instances is no longer sufficient. So we really needed more advanced statistics than someone like me can do in Excel. So we worked for quite a number of years on how do we analyze actually growth curves. If you look at growth analysis in plants, and a lot of the publications are actually from the 1970s, and it's only sort of now, in a sense, being rediscovered now that we have these non-destructive phenotyping methods where we do get time course series. So growth measurements and time course series analysis is something that we had to grapple with again especially if we want to do QTL analysis where you're usually plugging in one single number rather than a curve. So what we've come up with is really, we take the raw data, we use smoothing splines, and we look at different degrees of smoothing, so different degrees of freedom. And we also compare whether we smooth directly on the raw data or whether we might, for example, do a log transformation. Once we've identified the most appropriate smoothing protocol, we then, based on the shape of the curve, identify individual intervals or time points where there is something changing in the growth curve. And we then analyze those specific time points, and for those we can then do a full mixed model analysis so we can get very good statistics. There's also the option of doing longitudinal analysis. However, that does not give you the same statistical power, and it also doesn't give you those individual numbers that you can then use in a forward genetics and QTL or GWAS analysis. So just an example, this was from a study where we looked at salinity tolerance in 2 different rice populations, one indica, one OSH. You can see here the relative growth rate, and this is now using the spline smoothing and analysis rather than an exponential growth curve. And you can see the typical response of plants where relative growth rate declines over time. And in blue, you can see control, and in red, you can see salt. And pretty much each of those tiny individual lines is one individual plant. And from those, we could then extract different intervals. So we looked at intervals from 2 to 9 days after application, 2 to 6 days, 6 to 9, and 9 to 13 to try and capture different times in that response and in that growth response. And what you can see down here, for example, if we look at the ratio of relative growth rate in salt and relative growth rate in control, we do have a fairly good range and genetic variation within those populations. So yeah, that's certainly a lot more encouraging than what I had seen originally doing hydroponics with barley. From a different publication, again using rice, but this is Campbell's study. He was a student with Harkamal Walia at the University of Nebraska, so not too far away from you guys. And again, we used the same approach to look at then the genetic loci in rice. And what you can see here is just the Manhattan plot for the growth response. And here are just 2 examples, one for a loci on chromosome 3, one for a locus on chromosome 1, and looking at the 2 different alleles or SNPs at this location, you can see quite significant differences between the populations for either the A or the G allele here, or the C and the T at this specific location. So moving to the soil-based system, A meant we had a sort of more reliable growth response. Because we can do it at large throughput means we can do the numbers for a genetic screen, but also because we developed the statistical methods to analyze the data, we could then use it in a forward genetic screen to identify loci of interest. We then wanted to see, can we push it even further? So far in everything I've shown you, we've treated the shoot of the plant as one single object. But the question is, can we dissect it even further? And the results I'm showing you now is probably 3 to 4 years of work by very talented people in computer vision. Because the challenge we set them was, quite full on. We didn't want to work with maize because that's not a really big crop here in South Australia. We wanted to work with wheat or barley, which don't have the nice regular leaf arrangement that maize has, and obviously leaves are also a lot smaller. And we also didn't want to take hundreds of images per plant to do a 3D reconstruction because then our throughput would go right down. We wanted to use just 5 to 10 images maximum, so we can still do hundreds of plants every day. So we approached, they approached it similarly. So we start with the raw image. We do a foreground background separation to identify the plant, and then pretty much connect the path. So in pink here, you can see the individual segments that are identified as plant to connect them to one object. The blue frames that we use to keep the plants upright are rather annoying for the image analysis experts because they can mean breakage. So that's where we then have to sort of reconnect those segments. They then also use knowledge about what leaves can look like. So we know that such sharp angles like here in F are very unlikely, and that we have, more likely E is the correct path identified. Similarly, leaves are unlikely to go down and then back up. So we use some prior knowledge about what leaves look like and how they behave to identify the correct path. And we then also use the camera geometry to find points in 3D and finally track the leaves over time. So this is an example here where you can see color-coded the individual leaves that have been identified. And then from day to day, you can see the challenges that leaves move and plants move, but it still manages to find the same leaves. One challenge you can see already, once plants start to produce secondary tillers, at this stage we are not able to differentiate between whether a leaf is on the main tiller of wheat or barley or whether it is on a secondary tiller. We can only count sort of the order in which they appear. Nevertheless, our ability to measure individual leaf length and also total leaf length on the plant was really good, and we got super high correlation between manual measurements and the digital measurements up to about 12, 14 leaf stage. Then it becomes too complex for the approach we're taking. So we've used this approach to look at a biparental population in barley, and we screened both for 2D traits, so overall shoot size, absolute and relative growth rate. And then we also looked at 3D traits such as leaf length of, for example, leaf 4, or leaf number. And you can see here the results for Manda and Keel, which are the two parents of that population. And you can see that Manda here in blue has quite longer leaves, certainly leaf 4, whereas Keel has generally a higher leaf number than Manda. We then screen an entire population with about 240 recombinant inbred lines with salinity added to the soil, and we image daily to extract 2D and 3D measurements. And this is the overall population with control plants and the corresponding salt plants, and you can see the reduction in shoot growth overall. If we now look at the individual leaves in control and in salt, you can see that leaves 1, 2, and 3, there is little difference. That is because salt was applied when leaf 3 was almost fully emerged, and leaf 1 and 2 had already been fully emerged. Leaf 4 was sort of expanding under salinity, and you can see a reduction here in the length in salt compared to control. Leaf 5 onwards, you can see some are a lot longer than others, and that is because we then have the difficulty. Some are true leaf 5 on the main tiller, others might be leaf on the secondary tillers. So at this stage, we can only detect the order of leaves, but we can't necessarily tell whether they are on the main tiller or not. But you can see generally a slight reduction in length. We go further down, what you can also see is that, for example, here with the 7th and the 8th leaf, is that you have less traces, individual traces in salt compared to control. So not only is leaf length affected, but also leaf initiation. So salt stress plants has also less leaf and shorter leaves compared to the control plants. We then used all these traits. We extracted both the 2D and the 3D, both the primary traits as well as growth rates extracted from those. We looked at various time points, individual time points and intervals, and also at the control and the salt samples to do QTL mapping. We found 65 overall. Most have a very low lot score. With small effects. So it's multigenic traits, which is not surprising for something like salinity tolerance or growth in response to salinity tolerance. And we also, what was interesting, that we found more QTL that were for rates of change rather than absolute values on individual days. But something that is very important to remember, and that not only we have seen, but many others doing similar studies, is that the majority of QTL are transient. They appear and they disappear over time. So that's something important to keep in mind, especially when you're only doing endpoint analysis, is that up until that point, there have been a lot of QTL that appeared and disappeared. So just some key results. We found some of the usual suspects, such as photoperiod H1 or the flowering locus. So these are not surprising, given that we're looking at plant size and growth. We also found some related to, salinity and salinity transport, which is not unsurprising. We found a significant QTL on 3H, and that was for the length of leaf 4. So leaf 4 was the one that started to expand as we applied salt. That locus was only apparent on the second day after salt application and then disappeared again. And if we looked under that locus, there were 6 annotated genes. 3 weren't expressed according to published data. And the other 3, we had 2 expansins and 1 glucosidase, all of which are known to relate to cell wall modification and cell wall expansion. So the fact that they might be related to leaf elongation and leaf growth is quite sort of exciting, and certainly something that would be worth following up. Now, another example is a project that was mainly done by a PhD student quite a number of years ago, where we started with a very significant QTL for a sodium accumulation in the shoot that was actually identified in hydroponics. In barley, the candidate gene was a vacuolar pyrophosphatase. Back then, cloning of genes in barley was not that easy, so we actually, for simplicity for that student to go ahead, we started with the Arabidopsis version, AVP, so the vacuolar pyrophosphatase, and transformed that under overexpression with 35S into 2 different barley backgrounds, Golden Promise and a sort of local breeding line. And the idea originally that we had was that this pyrophosphatase would be responsible in sequestering sodium into the vacuole away from the cytoplasm so that it wouldn't be toxic to the cytoplasm and therefore increase tolerance. And we could certainly see that those plants grew a lot better. in salt. And Rhiannon Schilling then was sort of tasked in her PhD with trying to study those lines in more detail, also using growth analysis. So we did a greenhouse trial where we grew the plants, and these are the results at the end of the experiment. We did 47 days after planting, and at this point, salt was added right from the beginning to the bottom layer of the soil in the pots. And you can see that especially those two lines of the transgenics were significantly larger than the equivalent nulls of those lines. What was interesting when we looked at the growth rates, even though those transgenics are significantly larger, the growth rates weren't larger at all, neither in the early days nor in the later stages of the experiment, which was slightly puzzling. But the answer was really that the plants were already larger at 9 days when we started the imaging. So even though we thought that the vacuolar pyrophosphatase would increase salinity tolerance by sequestering sodium into the vacuole, what we found is that it actually increases early vigor, and there is now more and more evidence that suggests that that specific gene and protein have a role in early sugar metabolism and therefore also early vigor. So having that high resolution of imaging, we actually, had to rethink our understanding of the gene. If we had only looked at this data, we might have still suspected, yeah, that's because, it makes the plant more salt tolerant. However, the imaging and the early imaging clearly suggested, okay, this might be a vigor rather than a salt tolerance gene. We also took those plants out into the field, and as Francisco already showed, quite important to characterize your field site. So within the same field, we had a high salinity and a low salinity site over in Western Australia. And I have to admit, the high salinity was probably too high. We were rather pushing the plants there. What you can see from the image here, and also slightly reflected in the numbers, is that again, the ABP lines seem to be just slightly more vigorous and sort of bushier than the wild type plants. And yeah, as I mentioned, the salinity was probably a bit too high. Most of the wild type plants completely failed and failed to set any seed. However, the ABP lines managed to push through quite a number of them and actually did set seed. So they performed significantly better in this environment than the wild type ones. And both ABP1 and the wheat orthologs are now actually being tested as part of an Iweb project. So some of those sort of follow-up lines in wheat are now also being screened in the CIMMYT trials for the Iweb hub. A different example, this time in wheat rather than barley. So we did, again, a forward genetic screens in a double haploid population this time of Excalibur and Kukri. And again, we screen for growth-related traits, so that early response to salinity, but also some of the ionic traits, so how much sodium and potassium is accumulated in the shoot. And we identified quite a number of QTL, as you can see here. And what we then did is test lines that have the different alleles in that specific locus in the field to see what their effect was on yield. So first we looked at some of the ionic loci and how they affect leaf sodium or potassium accumulation. In bold is always the ones which are having the effect that we sort of hoped and anticipated, which was in this case a reduced accumulation of sodium or an increased accumulation of potassium with the ones of the beneficial allele versus the other allele. But you can also see that, quite a number of the field sites or years, that sort of prediction falls over. So there's clear evidence for G by E interaction here. If we then look at the grain yield results, we have here in the first column a QTL for a growth response, and that was beneficial in one of the sites and one of the years, but had quite negative impacts in others. Similar for the QTL that were identified for sodium or potassium accumulation. So really what that shows is that to do trait selection like that in the field, you really need quite a large number of fields and years to understand, okay, in which scenario and in which environment and under which conditions is it favorable, under which one is it a disadvantage. And that can depend on whether there is confounding effects with heat maybe, or with drought, or other environmental factors that year. Also, what was the overall yield in that year and then that site compared to other years. So in summary, both 2D and 3D imaging can be used for trait dissection in the controlled environment. What's important to remember is that a lot of the loci are transient, which is important if you just do an endpoint measurement. And we can then use those genetic loci that we identify under controlled conditions and test them for their influence on yield in the field. One important thing to remember is that phenotyping really is a team sport and requires many disciplines. So as a plant scientist, I'm actually in the minority in our team. We have statisticians, software engineers, experts in hyperspectral image analysis, obviously technical staff, electrical and mechatronics engineers, whole range of different disciplines really working together. And as Francesco already highlighted, in the field, there is a whole range of new tools coming online, both with UAVs or ground-based phenotyping. And the bottleneck in phenotyping is now certainly shifting more from the data acquisition towards the data analysis, because the data we generate is becoming more and more complex, and analyzing that at high throughput is becoming a bigger challenge. So coming back to that sort of controlled environment versus field. And I hope I could explain that, yes, even with controlled environment, there is value in doing that in the early research and pre-breeding pipeline, as long as you make sure that you then test things in the field to see whether they hold up or whether they completely fall over. And certainly with more and more field phenotyping tools becoming available, I think there is obviously increased capacity to phenotype directly in the field. And certainly with genomic selection becoming more and more popular in breeding programs, I think having really good phenotyping tools in the field will be critical. With that, I'd like to really thank all the wonderful funding bodies that keep our lights on, and a whole range of collaborators here in Australia and around the world. And if you ever want to come to Adelaide, this is the view from our campus. And that koala isn't a prop, he was actually sitting there taking in the view. So yeah, with that, I'd like to thank you all for listening and staying up on a Friday afternoon.